Hello, everyone, and welcome to today's webinar, Characterization of Lentiviral Vectors using Capillary Electrophoresis Platform Technology. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SciEx. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Aaron Shafer, Senior Scientist in the Analytical Methods Development Group within the Children's GMP at St. Jude Children's Research Hospital. Aaron, you may now begin your presentation.
Well, first of all, I just want to thank you for that brief introduction. Today, I will be talking about the characterization of lentiviral vectors using capillary electrophoresis as the platform technology. Before I get going, or before we dig into the talk, I did want to give you a little background some background about St. Jude Children's Research Hospital, which is where I work here. I work here at the Children's GMP, which is actually part of the hospital itself. And the main mission of our hospital is finding cures to treat kids, to save children from pediatric cancer. So the founder, Danny Thomas, a long time ago said that no child should die at the dawn of life. So it's the kids that are in the hospital that I see every day that really drive our mission and that keep us on point and motivated for doing what we do here at the Children's GMP. So we have a very large impact here at the hospital in that a lot of the products that are coming out of the research and development side are considered orphan products, and they would typically not find any interest in large pharmaceutical companies just because the scope or the number of prospective patients is too small for them to make that sort of investment. So it was decided that we would build our own children's GMP here on site, and we at the time we're the first pediatric cancer children's hospital, that is, to build our GMP on site. So some of the achievements and milestones at our hospital include the cure of hemophilia B using an AAV gene therapy approach. That was published in 2014. And I did want to make a mention that we don't only treat pediatric cancer, but we also are involved in other collaborations throughout the world with treating diseases that affect not only children, but also adults. So we also are a part of the World Health Organization's, we're a center of excellence for the World Health Organization for monitoring and coming up with seed stocks for bird flu to prevent avian pandemics. And then, as you see here, we've also had collaborations with patients in England for treating hemophilia B. So even though our focus is pediatric cancer, we have our research hands involved in a lot of different products that we manufacture here. Another advance include the cure of bubble boy disease using the lentivirus-based approach, which was published in 2019. Essentially, we were able to reconstitute a child's immune system where children are born without immune systems, and so we were able to treat them using the lentivirus therapy and basically generate a new immune system for them. In 2021, we published a study about treating children with neuroblastoma with one of the antibodies that we manufacture here at the GMP. And so using a combined chemotherapy monoclonal antibody approach, we were able to significantly reduce the mortality rate, i.e. the 3-year survival rate of these children who had these neuroblastomas and were not responding well to just the chemotherapy alone. And then, of course, back in the 60s, we started with acute with leukemia cancers in the 60s, where there was about a 6% survival rate, and we have that survival rate all the way up to around 95% now. So we've made a lot of progress in the last 50 plus years. All right, so with the background taken care of, let's talk about the agenda for today, for today's talk. First, we're going to go into a little bit about what a lentivirus, what does a lentivirus look like? What are the platform methods that we're going to look at today that will allow us to analyze these lentivirus products, which we'll talk about today, which includes the lentivirus proteome analysis. So we're going to discuss the titer determination by analyzing the P24 protein. We're also going to look at lentivirus protein profiling as well as the genome analysis. And within the genome analysis, we'll be able to look at the size of the genome within the lentivirus as well as evaluate the purity of the product. And then we'll finish with a summary. So lentivirus vectors. Lentivirus vectors are essentially a family of viruses that have single RNA in there, and they've been used for a numerous number of applications for delivering genes and stably delivering genes into patients that have been affected by a various number of diseases. So some of the advantages of using these viruses include your ability to transduce not only non-dividing cells but dividing cells as well. You can accommodate large transgenes all the way up to 10 kilobases, unlike the AAV viruses, where you're limited to genes that are less than 2 kilobases. There's low immunogenicity associated with these viruses. There's a broad tropism for the viruses, so you're able to infect a multitude of different cell types within the body. We also observe long-term robust transgene expression, both in vitro and in vivo. So these viruses do stably integrate into the genomes of the hosts. You can accommodate not only a single transgene, but you can express and stably integrate multiple transgenes. You're able to integrate with it, again, as I mentioned, you're able to integrate into the host genome, and that integration persists beyond, is stable so that when cells divide, you maintain the presence of that transgene. And then last but not least, it's stable ex vivo. So some of the applications include introducing new genes, for example, like introducing the gamma gene into patients that have this bubble boy disease, silencing of gene expression using the delivery of single hairpin RNAs, delivery of tumor-specific T-cell receptors so that you can hone in and have your immune system go after tumor-specific antigens, and then also the delivery of chimeric antigen receptors, where you're stably transfecting the T cells and targeting the tumors via these chimeric antigen receptors that are on the T cells, which will allow them to recognize the tumor cells. So the proteome of the lentivirus is comprised of 5 important structures and some non-structural proteins. All of them play important roles in the life of the cycle of the lentivirus. For example, the envelope proteins, the glycoprotein complexes, enable the virus to attach and diffuse the target cells and initiate the infectious cycle. For the capsid proteins, the quantification of the P24 protein is usually used for lentivirus titer determination. So here we've developed a separation-based method for lentivirus protein analysis with 2 purposes. First, to provide the accurate titer determination of the lentivirus sample based on quantitating the separated P24 capsid protein without any interference of the other proteins in the sample. At the same time, the well-separated protein profile could be utilized for lentiviral protein purity assessment. So first we'll look at titer determination by evaluating by correlating P24 protein to the titer of the lentivirus. So the workflow started with an easy and efficient two-step sample preparation protocol, which included disassembly of the lentivirus particles using SDS, and then covalent attachment of a fluorophore to the virus particles, or to the disassembled proteins, labeling the proteins and making them fluorescent. The resulting beryllium dye-tagged lentivirus proteome is then loaded onto the PA800 plus pharmaceutical analysis system, which is what we have here at St. Jude, or you can load those onto other CE systems. The resulting electropherograms is shown in the figure on the right over here. So you can see you get a whole array of different proteins eluting in the electropherogram. Essentially, in this electropherogram, there are over 20 proteins. In this lentivirus protein profile, the peak corresponding to the P24 protein is determined by spiking the lentivirus sample with a commercially available P24 standard. In the figure on the left, the trace on the bottom is the original separation trace of the lentivirus sample with the concentration about 1 × 10⁹ transducing units per mil, TUs per mil, while the 6 traces above the bottom one are the same lentivirus sample with spiked in with increasing amounts of the standard. The conclusion that we can draw from the figure on the left are the peaks in the circle with increasing area along the increasing P24 spiked in is identified as P24. So essentially the spike protein allowed us to identify the P24 protein within our sample. We also can see that there's excellent reproducibility of the protein profile. If we look at the other separated peaks in the electropherograms, even the various amounts of P24 in the samples, proving that the quantitative variation of the P24 protein content did not affect profiling repeatability. The resulting profile can be readily used for batch-to-batch reproducibility and protein purity assessment of the lentivirus samples. Also, by plotting the peak area of the identified P24 peak versus the amount of P24 spiked in, we could get a plot with very good linearity. As you can see, that the R-squared value is well above 0.99. Let's move on to the next slide here. Looks like we have 2 of the same slides. So here in this slide, we're evaluating the linearity and the LOQ of the standard. So this P24 standard was used to evaluate the linearity and determine the LOQ of this workflow by injecting increasing amounts of standalone P24 standard into the P800 plus system with the CE-SDS chemistry. So the figure on the left is the overhead traces of the P24 peaks with 2-fold zero dilutions, and the figure on the right is the corresponding linearity plot. Impressively, the plot covers 3 orders of magnitude, and we determined the LOQ to be around 8 nanograms per mil for the P24 standard. So what does the precision look like for injection to injection repeatability? For 6 consecutive injections of lentivirus sample, the relative standard deviation of the migration time and the peak areas are less than 0.2% and 0.2%. 0.91% respectively. So precision is excellent between injections of the same sample. So how about from batch to batch? That is, from one day to the next. What does the precision look like there? For 6 independent sample preps, the relative standard deviation of the migration time and peak areas are no more than 1% and 5% respectively. So the percent RSD is still quite satisfactory. The figure on the left is an example of the overlay P24 standard with different concentrations from a single preparation. The figure on the right is the average of the standard curves prepared and analyzed from 3 different days with the corresponding error bars on each point. So you can see from 3 separate days that we're still getting excellent reproducibility between preparations of the P24 sample. The quantity of the P24 protein of a lentivirus sample is determined using the average standard curve that was shown in the previous slide. After converting the P24 quantitation, or that is the peak areas, to lentivirus titer, the titer of the lentivirus sample is found to be 1.45, 1.45, and 1.46 times 10 to the 9th TUs per mil, or transduction units per mil, as was measured on 3 different days. So we have very consistent results. The numbers are consistent with the values obtained from the vendor as well, which the vendor obtained their TU data by using the ELISA assay. So this is basically a comparable method to the ELISA method that they used. So next, we're going to look at lenti-vector protein profiling. That is, what does the proteome look like in an electrophoregram for the lentivirus vector? So in this chart, what we're looking at is at the top electrophoregram trace, you see the LVGFP. That's the commercial standard that was, and then the following 5 samples are elute-dilute. They're called elute-dilute samples, and those samples were provided from our process development group here at St. Jude for ABCIX to look at. So what you can see here is that P24 has been identified in both the commercial, it's known where it elutes. So by using the commercial vector, we're able to identify P24 and the VSVG protein within our samples here from St. Jude. So in this slide, you can see there's a very strong correlation between the peak areas for the P24 protein as well as the VSVG proteins. And you would expect to see this sort of correlation when you effectively remove the free P24 during sample prep. If there was any free P24 in the solution, that could distort or cause that correlation to go away. So this indicates that there is no free P24, or that it's been eliminated from the profile during the sample prep step. And the last thing we're going to look at is genome sizing and impurity screening. That is, what does the genome, the RNA, look like from these lentivirus vectors, and what sort of impurities can we see? That is, nucleic acid types of impurities when we evaluate the nucleic acid content. So why would we want to look at lentivirus genome? What would be the importance when we're manufacturing lentiviruses? What sort of quality attribute is important for us? Well, knowing first of all that we actually have the genome in there is critical, because this is what ultimately allows the transgene to be passed on to the cells. So it's important to know that we have full-length genomes within the cells. And so there are situations where you could have incomplete RNA transgenes at the 3' end, and that could lead to failed reverse transcription in target cells and thus reduce transfection or transduction efficiency. Lentiviral vector genome integrity is one of the critical quality attributes too. So like I was saying, we need to make sure that we have a sufficient number of intact genomes so that we can have transduction of the transgenes. Also, we like to be able to evaluate residual host cell DNA and/or RNA. And with those sort of residual nucleic acids being present, you could have increases in the risk for innate immune responses. So purity is critical for these preparations, as well as evaluating the ratio of empty particles to full particles. So it's absolutely critical that we know that our genomes are of high quality. So this slide shows the workflow for genome integrity analysis. First, the nucleic acids are extracted from the samples and then analyzed on the CE systems, which could be either the Biophase 8800 system or the P800 system from AB-SciX. Results are shown in electrophoregrams in the panel on the right, which we'll get into more detail on the next slide. In this sample, the intact genome for the size has been determined to be around 6.1 kb and is well separated from the partial genome and other impurities. So this slide shows the results obtained for the analysis of four research-grade lentiviruses with different genome sizes from the commercial vendor. Each virus sample had a volume of 25 microliters, and the titer was 1E9 transducing units per mil. So we tried to extract the equivalent amounts of transducing units per sample. Peaks of the intact genomes are indicated by the arrows. RNA ladders are shown at the bottom as the bottom two traces, both in the pink trace and the orange trace at the bottom. So you can use those traces to size the actual size of the genome. This slide shows the assay repeatability results obtained with 12 injections of the LV cherry sample, which is shown in the left panel, and the LV GFP sample shown in the right panel. Peak profiles and migration times are very consistent between the injections. The RSD for the migration time for the intact genome peaks was less than 0.5% for both of the samples. So back to the samples that were obtained from St. Jude from our GMP. So this result shows results obtained for these. Samples were injected with a pressure injection and separated at a capillary temperature of 30 degrees Celsius. So with these samples, the amount of sample produced per sample, the number of transducing units would not normalize. So we would expect to see different genome sizes. And the only thing worth pointing out is that you can see there is some variability in the intact genome in terms of the peak shape. So what that means functionally in terms of how well each preparation transduces the cells is not currently known. So this slide shows results obtained in an in-depth analysis of the genome integrity of the LVGFP sample from the commercial vendor. The top panel shows the control results obtained without treatment of the sample before nucleic acid extraction. The middle panel shows results obtained when the samples were treated with either benzonase or DNase 1 before the nucleic acid was extracted. So Benzonase is a more non-specific nuclease that will cleave both any sort of form of RNA or DNA, versus the DNase one is much more specific for DNA. So the fact that those impurities that you're observing in the top trace are no longer present after treatment with benzonase or DNase 1 indicates that these are nucleic acid impurities that were present outside of the virus. And in the two bottom traces, you can see that the benzonase treated sample and the DNase treated sample look very similar, indicating that there's probably no contamination with RNA, because if there was, you would have seen RNA persist in the DNase 1 treated sample, and in which case the electrophoregram would look slightly different and have most likely a significant number more of impurities, RNA impurities. All right, so in conclusion, the CE platform technology demonstrates unique capabilities for quality attributes monitoring of lentivirus vectors. We've shown that there's a sensitive titer determination based on the precise quantification of P24 proteins with a 4 magnitude orders of detection linearity and an R square of greater than 0.99 and an LOQ of 8 nanograms per mil. With using this method, it's easy to assess batch-to-batch reproducibility in terms of titers as well as protein purities based on the evaluation of the resulting protein profile that we showed. We also observe a linear relationship between P24 and the VSVG proteins, indicating that the CE titer method is not measuring free P24, which is good. As I stated before, free P24 could confound results. The high sensitivity of the CE-LAF enables genome integrity analysis of the lentiviral vectors at a titer of 1 E9 TUs per mil without any sort of amplification step, providing high resolution of intact lentivirus genomes from various impurities with very good repeatability. So thank you very much for your time. If you made it all the way to the end, thank you for sticking with me. If there's any questions, or you would like to, if you had anything that you wanted me to clarify, please feel free to ask now. I'm available.
Thank you, Aaron, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, the CE method produced results similar to the ELISA method. What advantages are there to analyzing lentiviral samples by CE?
I think one of the main advantages, if you look at the range of the linear range of quantification, you have about for CE, it spans around 3 orders of magnitude. So your linear range goes from around 8 nanograms per mil all the way up to 1800 nanograms per mil. So you're working with a much larger range, which would mean you wouldn't have to look at trying to do multiple dilutions to try to figure out what concentration you should put your test sample at. So you have a much larger range for the CE method as compared to the ELISA method. Got about 100-fold or 2 orders of magnitude range in linearity for the ELISA method. So that range of quantification is more limited for ELISA. In terms of sensitivity, the CE method's got, as I mentioned, an LOQ of around 8 nanograms per mil versus around 4.6 picograms per mil for the ELISA. So the ELISA is a more sensitive method, but the range is more limited. So also in terms of sample preparation time, it's only you're only looking around 30 minutes to prepare your samples. For the CE method, you're basically going to be doing a buffer exchange into the matrix that's used to be is analyzed on the CE method, versus you've got multiple washes with antibodies and an amplification step with the ELISA, so that's going to be around 90 minutes. So just the front end sample preparation time is shorter. For CE, in terms of throughput, you know, with ELISA you're going to have a higher throughput because you can scan a 96-well plate relatively quickly, but with CE you're going to, again, you're looking at probably around 50 to 60 minutes per sample for analysis. So does that answer the question for you guys?
I believe so. So let's see. Our next question is, the protein profiling method demonstrated a linear correlation between the P24 protein and the VSVG protein. Is this an attribute that you will track in your process?
So I spoke with my process development director, who is in charge of manufacturing or developing a lot of scaling up processes for manufacturing our lentivirus vectors, and he said that there's quite a bit of variability in the VSVG expression between preps, so that it doesn't always correlate from batch to batch. You don't always have that same one-to-one relationship or linear relationship. So currently we're not using that in terms of using it as a quality attribute for our products that we're manufacturing right now, lentivirus products.
All right, thank you. Next question. The intact genome assessment reported a wide range of encapsidated RNA in the lentiviral particle. Will this workflow be used to optimize manufacturing conditions?
I think this method actually holds a lot of promise. We are not currently using it. This is a rather novel method, actually. So we're going to need to further evaluate whether there's a really good correlation between the genome content and the actual infectious titer. So I think we would need to make several standard curves from different batches and then calculate the infectious titers using the nucleic acid pCarry as a way to back calculate that.
All right, thank you. Next question. Was the initial titer determined via ELISA, and this method being the secondary proof of it, or is it strictly a CE method overall?
So if you look at the presentation, you can see that the plot has peak area of P24 versus the concentration of P24 protein, micrograms per mil. So in order to go from micrograms per mil to titer, you would use that conversion factor. So basically 1 nanogram of P24 is equivalent to 1.25 times 10 to the 7th lentivirus particles. We double check with Ting on this. We generated this data in our calibration, but I believe that was a direct conversion from the micrograms to virus particles, not actually using the P24 titer data from the manufacturer as a way to get that total particle count. So we need to double check that.
All right, thank you. Next question. Is the same matrix used for protein and RNA separation?
I guess we need clarification. Do you mean the matrix in terms of the gel that's inside the capillary, or how we treat the samples? Are you talking about the matrix inside the capillary? I wanted to clarify that.
I'm not sure. Maybe, hopefully, the person that submitted this question will follow up.
Sure.
Possibly.
Let's
go ahead.
Yeah, let's just assume we're talking. I'm going to assume that he's talking about what actual capillary or analysis method we're using? And the answer would be yes. It's an uncoated capillary that has the gel matrix inside of it, and that's used for both RNA and protein analysis, if that's what he's referring to, or the person's referring to.
Okay. All right. Thank you. Yes, came in. Yes, inside the capillary.
Yep. That would be a yes.
All right. Great.
Thanks.
Next question. What is the signal to noise ratio for the LOQ?
I did not calculate that, so I can't give an answer for that.
Okay. And the next question, why is the genome not affected by benzonase or DNase treatment?
It's protected, encapsulated within the virus, so it would not be anything, only residual RNA or DNA outside of the virus will be impacted by treatment with those two enzymes.
All right. Well, we will wrap up with that final question. Thank you again, Aaron, for your time today and your important research. We would also like to thank LabRoots and our sponsor, SciEx, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.